Okay, I'll start in like 2, 3 minutes. I'll start in like 2, 3 minutes. Right, so I'll share my slides. So this is the second live session of this course Structural Biology. I'll be discussing the lectures of week 2 and I will end by discussing some sample. Just 1 second. Yeah. Yeah, so this is the second live session, and I'll be discussing the lectures of week 2 and then discuss some sample questions. So this was the class that was just 1 second. I think if you can. Right. So I'll start the class. So this class is all about amino acids and proteins. As you know, amino acids are the building block of proteins. Now, the first protein structure to be determined at high resolution was myoglobin. And you can see here myoglobin consists of a number of alpha helical structures. In this class, we'll be discussing all about different secondary structures as well. Now, this is a polymeric structure made up of amino acids. And the basic structure of an amino acid is you have an alpha carbon to which there is an amino group NH2 and a carboxylic acid COOH. There is also a hydrogen and a R group. And this R group can be of 20 different types, forming the 20 naturally occurring amino acids. So here we'll just quickly go through the 20 naturally occurring amino acids. Just 1 second. Just reduce this. Yeah. So first we have the small amino acids that are glycine and alanine. So in glycine, there is no R group. The R group is another H. So glycine is the smallest amino acid. Then there is alanine where the R group is a simple methyl group. So these two are small amino acids. Then you have the nucleophilic amino acids, serine, threonine, and cysteine. So in serine, the R group is a CH2OH. Threonine is the same as serine, except there is an additional methyl group. And cysteine, the R group is CH2SH. So in these three, you have a nucleophilic atom, oxygen or sulfur, which has lone pairs. And so these form nucleophilic amino acids. The next class of amino acids are hydrophobic amino acids, in which the R group are simple alkyl carbon chains. So in valine, there is an isopropyl chain. In leucine, there is an isobutyl chain. In isoleucine, there is a secondary butyl chain. In methionine, there is a CH2CH2SCH3. So there is a sulfur, but because of the plus I effect of the methyl group added to sulfur, the effect of sulfur is reduced and it acts as an alkyl chain mostly. Proline is a very special kind of amino acid where you have the alpha, beta, gamma, delta, not as a straight chain, but as a cyclic structure. So in proline, there is a 5-membered ring cyclic structure, and it is the only cyclic amino acid. So these 5 form the hydrophobic amino acids. Then there are 3 aromatic amino acids where there is an aromatic group in the side chain. The simplest is phenylalanine with a CH2PH group. Then in the para position of phenylalanine, if an OH is added, it forms tyrosine. And instead of the normal phenyl group, if there is a indole group, then it forms tryptophan. Tryptophan is the amino acid with one of the largest molecular weights. You can see it is 186.21. There are 2 acidic amino acids. One is aspartic acid with a CH2COOH side chain, and one is glutamic acid where there is another additional CH2 group. So CH2CH2COOH. So in addition to the backbone acidic group attached to the alpha carbon, the acidic amino acids have an additional acidic group attached to the side chain, and so they are called the acidic amino acids. There are two amide amino acids where the side chains are an amide. So in aspartic acid, if instead of COH, you have CONH2, that forms asparagine. And in glutamic acid, if instead of COH, you have CONH2, that is glutamine. So here, because the side chain has an amide group, these are the two amide amino acids. And finally, there are three basic amino acids. One is histidine, where the side chain has a CH2 attached to a imidazole group. And here you can see the nitrogen has lone pair, so it can accept a H plus, thus behaving as a base. In lysine, you have a long alkyl chain, CH2, 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 ending in a NH2. Again, because of the lone pair of N, it can accept H plus and behave like a base. And arginine has a slightly more complicated structure where there is CH2, CH2, CH2 attached to a guanidinium group. This kind of group where three NH groups are attached to a carbon is called a guanidinium group. And here also, because of the lone pairs of the nitrogen, it can accept H plus and behave as a base. So these three amino acids are the basic amino acids. So you can see each of the 20 amino acids has a 3-letter code, like for alanine it is ALA, and a 1-letter code which is A. So it is important to remember the structure of the 20 amino acids, the 3-letter code and the 1-letter code. Now we will be discussing about different properties and how these amino acids form the proteins. So as you know, there are 4 elements which make up 98% of all biological matter. Carbon makes up around 18%, oxygen 65%, hydrogen 10%, and nitrogen only 3%. So these are quite light atoms. So these light atoms are what is most abundant in making up all biological matter, because small atoms have stronger binding between them. Now we know carbon has 6 electrons. So if you write out the electronic configuration of carbon, so this is the 1s orbital, this is the 2s, and this is the 2p. So the 6 electrons can be divided in 2 electrons in 1s, 2 electrons in 2s, and 2 in 2p. This is the ground state of carbon. In the excited state of carbon, 1 electron in the 2s is promoted to 2p. So now you have 4 unpaired electrons. And then there is a hybridization between the 2s and the 3 2p orbitals, that is which forms the sp3 hybridized orbitals. Now each of these 4 unpaired electrons can pair with 1 electron from one. So there are total 4 electrons from 4 hydrogens which are pairing with each of these unpaired electrons. That will form the methane molecule CH4. So you can see here 1 2s and 3 2p are combining to form an sp3 orbital which has properties of both s and p, and they are oriented in a tetrahedral orientation. And this has this is basically the hybridization of all the amino acids, the C alpha of all the amino acids as well. So if you consider this as C alpha is attached to one amine group, one COH group, one H and one R group through one sp3 orbital each. Now we'll look into electronegativity. The electronegativity is the tendency of an atom in a molecule to attract the shared pair of electrons towards itself. So if the atoms in a functional group have notable difference in electronegativity, then the functional group will be highly polar and thus it can participate in reactions. So if you have similar groups like CH3, CH3 or OO, they have the same electronegativity, so there is no charge separation or polarity. Thus the dipole moment is 0. But if you have a polar bond like HCl, the electronegativity of Cl is greater than H, so it will pull electrons towards itself, gaining a delta minus charge on the chloride. The hydrogen will then gain a similar delta plus charge. So this leads to the presence of a dipole moment in the HCl molecule. Now, as you go from hydrogen to carbon to nitrogen to oxygen, the electronegativity increases. And if you have a molecule like water, that is oxygen attached to two hydrogens, since oxygen has higher electronegativity than hydrogen, it will pull the electrons closer towards itself and gain a delta minus charge, whereas both the hydrogens will get a delta plus charge. So this is how there is charge separation in a water molecule. And because of this charge separation, different water molecules can associate through hydrogen bonds, where the delta plus on the hydrogen is forms a polar bond with the delta minus on oxygen, and these are called hydrogen bonds. So these hydrogen bonds in water leads to a cluster structure which forms the ice, and when heat is provided to the ice in order to melt it, hydrogen bonds gradually break till eventually all the hydrogen bonds break and the water forms a gaseous state. So it is due to this difference in electronegativity that hydrogen bonds are forming. And we will later see in this class how the hydrogen bonds lead to formation of tertiary and secondary structure in proteins. So one classification of amino acids we saw was based on whether the side chain was acidic or amide or basic. Another classification is known as the hydropathy classification. The hydropathy index of an amino acid is a number representing the hydrophobic or hydrophilic properties of its side chain. So there is a number. We don't need to know how the number is calculated, but larger the number, more hydrophobic the amino acid. So you can see the most hydrophobic amino acids are isoleucine, which has a hydropathy of 4.5, and valine, which has a hydropathy of 4.2. And this makes sense because if you go back and look at isoleucine, you can see it has a very hydrophobic side chain, which is just composed of CH2 groups. And similarly for valine. And the most hydrophilic amino acids are lysine and arginine, which have minus 3.9 and minus 4.5 hydropathy. And again, if you go back, you can see lysine has a NH3 plus and arginine is also charged. And so these make sense that these are hydrophilic. So why this is important is because when a protein is folding, the hydrophobic amino acids tend to be in the internal region of the protein with respect to its three-dimensional structure, while the hydrophilic amino acids are more towards the protein surface. Because you know the protein surface is exposed to different solvents like water, which is so water forms most of the body's fluids. So the protein outer surface will be exposed to water. So if the hydrophilic amino acids are on the protein's outer surface, that will help to stabilize the protein. That is why the hydropathy classification is important, because it will determine the three-dimensional structure of the protein as to which amino acids will fold to be in the internal region of the protein and which will remain exposed to the solvent. Another classification of amino acids is the volume classification, where you have the very small amino acids, which are glycine, alanine, and serine, and then gradually increasing sizes till you have the largest amino acids, which are the aromatic amino acids, phenylalanine, tyrosine, and tryptophan. Tryptophan, we have already discussed, is the amino acid with the largest molecular weight. So these are the different classifications of amino acids. Now, amino acids other than glycine have a chirality in them. So in the last class, we saw that we will call a molecule chiral. So if a carbon is attached to 4 different groups, then that carbon is said to be chiral. Now we know that in glycine, the R is hydrogen. So in case of glycine, the alpha carbon is not attached to 4 different groups because 2 of the groups are hydrogen. So glycine is the only non-chiral amino acid. All other amino acids have a chirality. And chirality, we saw in the previous class, chirality means that the molecule and its mirror image is not superimposable. So this chirality exists in 19 out of the 20 amino acids, and the amino acid can exist in two enantiomers, L and D enantiomers. That is one property of amino acid, which is the chirality. Another property is the pKa. So when you have an amino acid with the amine and the acidic group, then in a neutral environment, it makes sense that the H plus of the acid loses the H plus and the amine group captures the H plus. Because in the same molecule in close proximity, you have acidic and basic groups. So the acidic group loses H plus and the basic group gains the H plus. The net charge is still zero because you have one positive and one negative charge which cancels out, and the net charge is zero. This state of the amino acid is called a Zwittered ionic form. Now, as the pH is increased till the pH, say pH around 2, which is acidic pH, then so acidic pH means the solvent is providing H plus, then this CO minus will also be protonated and become COH. Then the molecule will just have a single plus charge and the net charge will be +1. Or if you have an alkaline environment with pH around 9 or greater, so in alkaline environment, the solvent will take away this H plus. So NH3 plus will become NH2. So in alkaline environment, the net charge will be -1 due to the CO minus group. So depending on the pH of the environment, the amino acid can have a zero charge, which is the Zwittered ionic form, or it can have a +1 or ±1 charge. Now, this is for amino acids where the side chain does not have any charge. So here the same thing is shown. You can see that in pH of around 2, there is a net +1 charge, and pH of around 9, there is a net -1 charge. Now, the point at which the net charge is zero is called the isoelectric point. And how to calculate the isoelectric point is that, so the state where the net charge is zero, surrounding that state, there are two pKa's. So there is pKa1 and pKa2. The average of these two pKa's forms the isoelectric point. So you can see here an example is, we'll start with alanine. So in alanine, you have the alpha carbon attached to a CH3. And if it is we'll consider the fully protonated state first, where the NH2 is protonated and is NH3 plus, and the carboxylic side chain is COH. So there is one pKa, which is for the carboxylic acid, which is 2.34, and one pKa for the NH3 plus proton, which is 9.69. The average of these two pKa's gives you a number which is called Pi, or the isoelectric point. So often you can get questions where you are asked to calculate the Pi. And how the isoelectric point is calculated is by taking the average of the two pKa that flank the net zero charge. So in alanine, there is no charge on the side chain. So you just have one positive charge, one negative charge, and zero charge. So around the zero charge, there are two pKa's, which when you take the average, you get the isoelectric point Pi. Now, what if you have aspartic acid? In aspartic acid, the side chain also has a charge. So we look at the different forms in aspartic acid. When everything is protonated, that is at the most acidic pH, the NH3 plus has a positive charge, so the net charge is +1. Slowly, as you increase the pH, the first H plus lost is from this main chain COH, and the molecule has a net charge of 0 because this plus and minus cancel out. As the pH becomes more and more basic, the side chain H plus is also lost, and now the molecule has a net charge of -1 because -1 and +1 cancel and a net -1 charge is left. And at very high pH, very alkaline pH, all the H plus is lost and the molecule has a net charge of -2. Now, in order to calculate Pi, we have to take, so there are three pKa's, one for the main chain COH, one for the side chain COH, and one for the NH3 plus. What we have to do is we have to see which is the zero charge form, and we have to take the 2 pKa on either side of the zero charged form. So 2.1 and 3.9. Taking the average of that will give you 3, which is the isoelectric point in aspartic acid. Similarly, if you see lysine, so this is the case where you have an acidic side chain. So the side chain has a COH group. Now, if the side chain has a basic group like lysine here, so now also there are 3 pKa's, one for the main chain COH, one for the main chain NH2, and one for the side chain NH2. Here we have to take, so when the first COH is lost, this is lost, then there is a net positive charge. But when, so here you can see there are 3 pKa's, and for this case, in order to find the isoelectric point, we have to take the average of the second and third pKa, that is 8.95 and 10.53. The average is giving you 9.74. That is the isoelectric point of lysine. So like this, the isoelectric point can be calculated. Now we will look into something called polyacrylamide gel electrophoresis, or PAGE. What happens is, suppose you have a protein which is folded. Now, in the folded protein, there can be different charges, like plus charge wherever there is basic amino acid, minus charge wherever there is acidic amino acid, and there can be disulfide bonds. So you can see SS disulfide bonds. Now, when you react, so there are two different proteins here. When you react the protein by 2-mercaptoethanol, the disulfide bond is reduced. So the SS bond is broken and it becomes SH-SH. So the disulfide bond is reduced. Then you are adding SDS, that is sodium dodecyl sulfate, which is a detergent. So it is a large molecule with a negative charge. And what this molecule will do, it will bind to different regions of the protein and it will lead to unfolding of the protein. Because there are so many negative charges right now, they will repel the interactions within that were holding the protein together, and it will lead to unfolding of the protein. So after treatment by 2-mercaptoethanol and SDS, the protein will be completely unfolded and there will be different SDS molecules attached to the surface of the unfolded linear protein. Now, depending on the size of the protein, so this is a large protein, there will be many SDS molecules attached to it. Or if there's a small protein, there will be less number of SDS molecules attached to it. Now, there is a setup like this where you have different, you can see this dent here. This is where it's called a well. So in that well, the sample is put. So suppose you have a mixture of, say, 5 proteins. So you put one protein in each well and there is a buffer solution. And a potential difference is applied across the setup. So you have a positive charge below and a negative charge above. Now, because of the SDS, the protein molecule has a net negative charge. So the protein molecule will travel down towards the positive charge. And if the protein molecule is large, it will move slowly. So you can see this is a large protein. It will move slowly. So it has, after a certain time, it has only moved this much. But this is a small protein, so it will move much faster. So after a certain time, it has moved a larger distance. So using this method of polyacrylamide gel electrophoresis, or PAGE, a mixture of proteins can be separated based on their molecular size or charge. So this is a method for separation of proteins. Another concept was discussed in week 2, which is the effect of chirality of protein amino acids on the bacterial cell wall. So peptidoglycan or murine is a polymer consisting of sugar and amino acid that
Hello, ma'am. Am
forms.
I audible?
Uh-huh. Yeah, you are audible.
Can you go back to the previous slide, please?
Hello?
Hello, ma'am. Can you go back to the previous slide, please?
Yes, please
Uh,
ask.
yeah, yeah. Here it says it's HDS, ma'am. What is the function of HDS in this acrylamide electrophoresis?
So SDS basically attaches to the surface of the protein. So when you have and SDS has a negative charge. So because of the repulsion, so suppose you have a proteins a folded protein like this, there are many interactions that fold the protein into the tertiary structure, right? SDS, what it will do, it will bind to different regions of the protein and it will unfold the protein. That is one thing. Another thing is that SDS also contributes in increasing the molecular weight of the protein, so it can be separated out while this electrophoresis is happening.
Okay, ma'am. Okay. Thank you.
Okay, so I was talking about the chirality effect of the amino acids. So basically, in bacteria, you have a plasma membrane that is formed of this polymer called peptidoglycan or murine. And this polymer is made up of different sugars, of two sugars. One is the N-acetyl glucosamine and another is the N-acetyl muramic acid. So these sugars occur in alternating pattern, and in between the sugars, they are connected by different amino acids. So there is a mixture of L and D amino acids connecting or cross-linking the sugars. Now, why the chirality? You don't need to remember the structure or anything, but this is an example of why the chirality is important. Actually, what happens is that here it's not shown, but there is another D-ala under this. So this is D-ala, there is another D-ala under this. So the D-ala structure is very similar to the penicillin structure. And what penicillin does is that we know penicillin kills the bacteria, but how it kills the bacteria is that, so in the native structure, there is D-ala. But if penicillin is added, because the structure is so similar, you can see D-ala is forming this kind of a structure, and penicillin here also has a very similar structure. It will compete with the D-ala and it will insert itself into the cell wall. And what that will do is, because penicillin is a non-native molecule, if it inserts itself into the cell wall, it will cause disintegration of the cell wall, and that helps in killing the bacteria. So that is why the chirality is important, because it is D-ala, the penicillin, this structure of penicillin is working as an inhibitor, where it is replacing the D-alas in the cell wall. But if this was L-ala, then another structure, penicillin would not have worked. So depending on the chirality of the amino acid, different drugs are chosen. So as you know, most of the naturally occurring amino acids in your body are L amino acids. So if you are taking a medicine that has, if you are taking a peptide based drug, that also has to have L amino acids. Otherwise, it will not work in the body. But in bacteria, you can see there are D amino acids. So to kill the bacteria, penicillin is working. So the chirality of the amino acids actually determines what drug can be used. Now we'll go into the peptide bond. So basically, when two amino acids come together, a molecule of water is lost and it results in a CONH amide linkage, also called the peptide linkage. Now there is a resonance in this linkage. So you have CONH and NH has a lone pair. So this lone pair can be donated and this double bond can break to give a partial double bond structure here. So because of this kind of resonance, the peptide bond is planar. So you can see the peptide bond, so here the two groups shown by the blue circles are on opposite sides. So this is a trans peptide bond. And if the two groups are on the same side, it is a cis peptide bond. So in general, the trans peptide bond is sterically preferred, because in cis peptide bond, you can see there is a steric clash. So interconversion between trans and cis, if it was a single bond, it would have been freely allowed. But because it is a double bond, this free rotation is restricted. And so the trans and cis, so you can see the rotation around this partial double bond is 88 kilojoule per mole. And so the trans and cis are separated. They are not interconvertible into each other.
Ma'am, please explain the trans and cis bonding once more. I
Okay,
didn't quite
so you
understand
can
it.
see, you can see this peptide bond, right? CONH. In CONH, so this is one C alpha, this is another C alpha. If the two C alpha are on and this is the double bond. If the two C alpha are on opposite side of the double bond, that is trans. And here the two C alpha are on the same side of the double bond. So that is cis. It is like normal trans and cis that you, in a double bond, if you have two, this is trans and this is cis.
And ma'am, what about the partial double bond character?
Yeah, so you can see, so this is one structure. Now, if the lone pair is donated here and this bond breaks, then this is the structure. So these are two resonance structures. Now, resonance structures, you know that it will not completely be in one, it will not completely be in two. So this bond, you cannot say it is a full double bond, it is a partial double bond. Because in one resonance structure, there is double bond, in another resonance structure, there is no double bond.
That means that it will convert between this and that.
Yes,
Structure.
it is existing between 1 and 2. And so this bond that you can see, that is why it's called a partial double bond, because one structure has full double bond, one structure has single bond.
That means sigma bond is forming here, no?
Sigma is already there, pi is forming, no? The double bond is pi.
Oh, right. My bad.
This
Sorry.
is pi.
Okay, ma'am. Okay, ma'am. I have
Okay.
understood that.
So in protein structures, you have different levels of structure. The primary structure is simply the amino acid sequence connected by peptide bonds. Then you have secondary structure and tertiary and quaternary. We will go into each of these. So secondary structure is basically where there are different kinds of secondary structures like the alpha helix or beta sheet. We will go into each of these in the next slide. So the alpha helix can be of two types. One is the left-handed alpha helix, another is the right-handed alpha helix. Now, all natural alpha helices are right-handed. This is because if you see the left-handed alpha helix, the CO and the C alpha and substituents on C alpha are very close. So there is a steric repulsion between them. So energetically, the left-handed alpha helix is not preferred. But if you look at the right-handed alpha helix, you can see the CO is pointing up and the C alpha and its substituents are pointing down. So sterically, the right-handed alpha helix is preferred because there are no van der Waals clashes between the substituents on C alpha and this carbonyl oxygen. Now, in the right-handed alpha helix, it has some properties. One is that there is a hydrogen bond between I and I plus 4 amino. So let us first label, how are we labeling the I and I plus 4? So this carbonyl, let us label this as I. So after carbonyl, there is NH, C alpha. Wait, I'll label according to C alpha. So if I call this C alpha as I, C alpha is attached to CONH. Next C alpha is labeled as I plus 1. Again, you follow to the next C alpha, which is labeled as I plus 2. And next C alpha is I plus 3. And again, the next C alpha is I plus 4. Now, if you can see, the NH of I plus 4 and the carbonyl oxygen of I are close together and they form hydrogen bonds, where the distance between this N and O is around 2.5 to 2.7 angstrom. So the alpha helix forms in such a way that the I and I plus 4 residues are brought close together, and there is a hydrogen bond between the CO and NH of I and I plus 4. Now, in one turn of the helix, it was found to have 3.6 residues, and per turn of the helix, the alpha helix is rising by 5.4 angstrom. That is called the pitch of the helix. Now, when you have multiple amino acids, each amino acid, there is a charge separation with oxygen having a minus charge and hydrogen having a plus charge. So there is a dipole moment in each amino acid. So suppose the dipole moment we are taking from plus to minus this direction, again from plus to minus this direction. So like that, each amino acid has a dipole moment. So the helix will, and since all the dipole moments are in the same direction, the helix will have a net dipole moment. And you can see the net dipole moment is around minus 0.7 to minus 0.5 dBs. That is called the macro dipole of the helix. Another thing is that if you look at the helix from the top, then this is called the wheel depiction of the helix. So suppose I start from this serine. Then the serine is connected to an alanine, which is connected to a histidine. Like that, if you go from amino acid to amino acid, from the top, you will get a kind of circular arrangement, and this is called the helix wheel diagram. And in this diagram, you can see that when you have, suppose I'm starting from serine to alanine to histidine to glycine. So 4 amino acids is forming around 1 turn of a helix. And the number of degrees that you turn per residue is around 100 degrees. So this is another representation of the helix. Now, how this representation helps us is that we can design helices that have different faces. So in this particular helix, you can see that the yellow residues are all leucine. So they are aliphatic hydrophobic. The blue residues are tryptophan, so they are aromatic hydrophobic. And these green residues are arginine, so they are positively charged. So you can design helices in such a way that suppose one face of the helix will be positively charged. So suppose you have a helix interacting with some other molecule, and one phase of, say this phase is exposed to solvent, and this phase is interacting with some other molecule. So in order to optimize this interaction, you can design the helix in such a way that the phase that is interacting with the hydrophobic molecule is hydrophobic, and the phase that is exposed to the solvent is hydrophilic, so that it is more stable. So the helix can be designed in such a way that different phases of the helix have different properties. Now, the alpha helix is the most common kind of helix, but there are 2 other kinds also. One is the 310 helix. So in alpha helix, there were 3.6 residues per turn. In 310 helix, there are 3 residues per turn. And in alpha helix, it was I plus 4 to I. In 310, it is I plus 3 to I. And the most rare kind of helix is pi helix, where the bond is I plus 5 to I. So because the pi helix, the bonding is at larger distances, the pi helix is larger in diameter. So there is a kind of cavity in the middle of pi helices. So pi helices can often be used as channels. So suppose you have to deliver a molecule through a channel, then the pi helix can be used. But it is a pretty rare helix to occur naturally. Now, in the alpha helix can associate in different ways. So these basically are the tertiary structures. So the alpha helix itself is a secondary structure, but if many alpha helices associate, they can form tertiary structures. So one kind of association is called coiled coil, where you have multiple alpha helices coming close together and they are held together by different non-covalent interactions. Another is the leucine zipper. Leucine zipper is one where there are only two alpha helices associating, but they associate in a very different kind of way. So the residues that are facing each other here, they are held together by hydrophobic interactions, and the residues on the surface, they are held together by ionic interactions. So that forms something called a leucine zipper, and the leucine zipper is a motif that is often found in DNA protein interactions. Again, you don't need to go into the details of these structures. This is just and giving you an overview of the different associations that alpha helices can form. So that was one kind of secondary structure. The other kind is your beta sheet. Now, the direction of beta sheet, so N to C terminal. So this is N terminal with NH, this is C terminal with CO. So the direction is from N to C terminal. So this is the direction of the top strand. Now the direction of the bottom strand is again from N to C terminal. So this is the direction of the bottom strand. Now if the top and bottom strands are in opposite direction, that is called anti-parallel arrangement, and it forms anti-parallel beta sheet. And both the top and bottom strand are in same direction. So here you can see N to C, same direction. N to C, same direction. Then it forms the parallel beta sheet. Now you can see that in anti-parallel beta sheet, CONH forming a hydrogen bond, again CONH forming a hydrogen bond. These are just C alpha substituents, so there is no bonding between them. Again CONH hydrogen bond. So in anti-parallel beta sheet, there are two kinds of cyclic structures formed. So if we start counting the number of atoms in the cyclic structure starting from this N. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this is a 10-membered ring. And the other cyclic structure, again, we'll start from this N. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So this is a 14-membered ring. So anti-parallel beta sheet has alternating 10-membered ring and 14-membered ring formed by hydrogen bonding interactions. Whereas if we count the number of members in the cyclic structure in the parallel beta sheet, again we'll start counting from this N. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So it is a 12-membered ring. And you can see the next is also a 12-membered ring. So in parallel beta sheet, it is only 12-membered rings that are formed by the hydrogen bonding interactions. So it has only one kind of cyclic structure. So these are just the basic structural features of the anti-parallel and parallel beta sheet. And the C alpha, so you can see these C alphas, the distance from one C alpha to the next C alpha is slightly greater in the anti-parallel, which is around 0.7 nanometer, and it is slightly lesser in the parallel, which is around 0.65 nanometer. Now about the beta turns. So if you have an anti-parallel beta sheet, they have to be connected in some way, right? You can't suddenly change direction. One which is going towards left, it cannot suddenly turn towards right. There has to be something which is enabling this change of direction. And that is where the beta turns come into play. So beta turn is a small structure made up of 4 amino acids, and it helps in a 180-degree turn in the direction of the chain. So you can, again, we'll count the amino acid by the C alpha. So this C alpha, we can call it I, which is labeled as 1. Then a peptide bond, then the second amino acid, which is I plus 1, another peptide bond, third amino acid, that is I plus 2, another peptide bond, and fourth amino acid, which is I plus 3. So there are total 4 amino acids in the beta turn. And when is a structure defined as a beta turn is when, so C alpha I, which is this one, and C alpha I plus 3, which is this one. when this distance is less than equal to 7 angstrom. That is the definition of the beta turn. And it is called a non-repetitive secondary structure. Because alpha helix, you have the I+4 hydrogen bonding constantly repeating, and it can extend. Beta sheet also, you have this kind of 14-10 hydrogen bonded ring or 12-12 hydrogen bonded ring, and it is a repetitive structure. But beta turns occur in isolation only when a turn is required in the protein. And so it is called a non-repetitive secondary structure. And there is hydrogen bonding in the beta turn. As you can see, this CO of I and NH of I+3 are close together. So there is hydrogen bonding. And there can be additional hydrogen bonding also, depending on whenever there is a carbonyl and NH coming close to each other, there can be hydrogen bonding, and these are stabilizing the beta turn. So the three kind of secondary structures we have seen are the alpha helix, beta sheet, and beta turn. Now, about the trans and cis isomer. What we just discussed a little while ago is that, so this is the amide bond. If we imagine this a double bond, if the C alpha are on opposite side, it is the trans isomer. And if the C alpha are on same side, that is the cis isomer. So between trans and cis, there is considerable energy difference. And so that free rotation cannot occur between trans and cis. But if one of the amino acids is proline, so proline, you remember, is a cyclic amino acid. In this case, in the cis isomer, there is steric interaction as you, so cis isomer, C alpha and C alpha are on the same side, and there is steric repulsion. But in proline, one special characteristic is that in the trans isomer also there is steric repulsion, because in the trans isomer, one is C alpha, and because the proline is a cyclic structure, the this is C alpha proline, C beta, gamma, and delta. So the C delta of proline comes close to the C alpha of the other amino acid, and so there is a steric repulsion between these two. So whenever you have proline, both cis and trans is destabilized by steric interactions. That is why the energy difference between cis and trans is reduced when you have proline. And because of this reduction in energy difference, conversion between cis and trans is easier in case of proline. So normally in other peptide bonds which don't have proline, the trans is very much dominant. Like the cis percentage will hardly be 5%. But when you have a proline, because the energy difference between cis and trans is much reduced, the percentage of cis increases. So cis will exist in around 30%, but trans is still the major, but cis, which was existing only around 5% in this, will become 30% here, because the trans is also destabilized by steric interactions. So now we'll come to the tertiary structure of the protein. So secondary structure we have covered. Tertiary structure is when, so imagine this is the polypeptide backbone, and there are certain non-covalent interactions occurring in the polypeptide backbone. So you can have hydrogen bonds, you can have hydrophobic interactions between alkyl groups, there can be disulfide bridges. So cysteine was CH2SH. If two cysteine come close together and the hydrogens are lost, it will form a disulfide bridge. Or there can be ionic bonds between an acidic and basic amino acid. So these kind of non-covalent interactions, which determines the three-dimensional structure of the protein, is what is giving stability to the tertiary structure. So you can have different kinds of proteins. One is the fibrous protein, where you don't have much tertiary structure. The protein is just existing in long chain. So whenever there is tertiary structure, the protein is folding. But in fibrous proteins, there is not much tertiary structure. So the protein is existing in long polypeptide chains. Comparatively, if you have a globular protein, the protein will fold and fold due to different non-covalent interactions and form a kind of spherical or globular shape. And these fibrous proteins are mostly insoluble in water, but the globular proteins are soluble in water because they fold in such a way that the hydrophobic residues are on the inside of the protein and the hydrophilic residues are on the outside of the protein. Because the polar and hydrophilic residues are on the outside of the protein, they are readily soluble in water. And some examples you can see, mostly of fibrous proteins are keratin, collagen, or silk. And globular proteins is mainly the hemoglobin or myoglobin that you have. So these are two kinds of proteins based on whether there is tertiary structure or there is no tertiary structure. And the final level of organization is the quaternary structure. Now, quaternary structure describes the joining of two or more polypeptide units. So this is a hemoglobin where you have, so this orange part is basically, this is a alpha helix. You have an alpha helix which is folded and held together. So the orange part is alpha helix and these are beta sheets. The blue is the beta sheet. So you have different units of the protein, and when they are held together by non-covalent interactions, that forms the quaternary structure. So you can imagine one, so there are four tertiary structures, two orange and two blue. Each of these are one tertiary structure. And when multiple tertiary structures are held together by non-covalent interactions, that forms a quaternary structure. So this is the highest level of organization in proteins. Now we'll talk about the dihedral angles. So any angle between four atoms is called a dihedral angle. So here we will define two important dihedral angles. We'll start from 1n, go to C alpha, go to CO, and then go to N. So basically rotation around this bond. This is the dihedral angle known as chi. And if you go from CO to N to C alpha to CO, that is rotation along this bond, that is the phi. These are the two important dihedral angles. Another is omega. So in omega is basically what we saw in cis and trans. So if you go from C alpha to CO to N to C alpha, that is rotation along the peptide bond, that is the omega dihedral angle. And so you can see omega is shown here where you have, this is the peptide bond. If the C alpha are on opposite sides of the peptide bond, it is the trans isomer where omega is 180 degree. And if the C alpha are on the same side of the peptide bond, it is the cis isomer with omega around 0 degrees. So these are the different dihedral angles in a polypeptide chain. Now, why these dihedral angles are important, we'll come to that. You can have dihedral angles in the side chains also. Like suppose we have the lysine side chain. So if you start from CO, N, C alpha, C beta. So this is CO to C alpha to C beta to C gamma. This rotation is called the chi dihedral angle. So your side chains can also have different dihedral angles. And here you can see three orientations of the chi dihedral angle. One which is called as G minus. In G minus, the CO and the side chain substituents are close to each other. And then is T. In T, the CO and side chain dihedral angles are opposite to each other. And there is G plus. So you can have different, again, we don't need to go into the details of this. These are just different dihedral angles in the side chains of the amino acids. But the important ones are the phi and chi dihedral angles we talked about here. Now, why these are important, we'll go into that. There is something called Levinthal's paradox. Basically, imagine a polypeptide having 100 amino acids. So 100 amino acids will mean 98 peptide bonds. Now, with each peptide bond, you have an associated phi and chi. So 99 peptide bonds will mean 198 different phi chi angles, because each peptide bond has one phi chi. So you have so many different phi chi angles. If you imagine that each side chain can exist in 3 conformations, then the total number of possible conformations of all the residues is 3 to the power 198. Now, if the polypeptide has to go through all these conformations before it can fold into the final native protein, then the time taken to fold one protein will be much greater than the life of the universe, which is not possible. So that means that all phi chi angles are not possible to be adopted. There are certain phi chi angles which are allowed in peptides. This you can also understand by this steric van der Waals graph. So when two atoms are coming close to each other, slowly the attractive forces increase till at a point the attractive force is optimized. If you bring the atoms closer than that, then there is repulsive forces which act and the net van der Waals force becomes repulsive. So again, what this says is that all phi chi angles cannot exist, because in some phi chi angles, there will be a large amount of repulsive forces between the atoms. So this is what Ramachandran did. He took the model of a dipeptide and he calculated that which phi chi angles are allowed and which phi chi angles are sterically disallowed. That gave rise to the Ramachandran map. So in Ramachandran map, on one axis you have phi, on the other axis you have chi. So most of the phi chi angles are, you can see this entire white region are the phi chi angles which are not allowed. Because if those phi chi angles are adopted, there is too much steric repulsion in the molecule. So only very small regions are allowed. And you can see this red region is the alpha R, meaning the right-handed alpha helix. This red region is the beta sheet. So when the polypeptide adopts phi chi values in this range, then it forms a right-handed alpha helix. If the polypeptide adopts phi chi angles in this range, it will form a beta sheet. So that is how the Levinthal's paradox was explained. The Levinthal's paradox was saying if all the phi chi angles had to be sampled, then it will take too long for a protein to fold. What Ramachandran found is that all phi chi angles cannot be sampled because they are not sterically allowed. So only between the allowed phi chi angles the protein has to sample in order to find the phi chi angle where all the interactions are optimized, and that will be the native state of the protein. So this is the general Ramachandran map for 18 of the amino acids. There are two exceptions. One is glycine, one is proline. In glycine, you know, the R group is hydrogen, so the steric interaction is very low. So in glycine, a much larger region is allowed. So you can see these are all disallowed in case of other amino acids because they have a side chain substituent. But in glycine, the side chain is hydrogen. So a much greater region of phi chi is allowed. And the other exception is proline. Now, proline you can see is a cyclic amino acid. So in cyclic amino acid, free rotation is not allowed. The angles in the cycle are fixed. So in proline, a lesser region is allowed. You can see this region is completely not allowed in proline. And so that is why proline also has a different kind of Ramachandran map compared to the other amino acids where the side chain is not cyclic like this, it is extended. So glycine and proline are the exceptions to the standard Ramachandran plot. Now, basically, in Ramachandran plot, you can have something called a dynamic Ramachandran plot. So you know that molecular dynamic simulation of a protein is basically where you allow the protein to move, and eventually it will adopt a state that is stable for the protein. Like you can here different states of the protein are shown, and it is being plotted on the Ramachandran map. So initially the protein started here, and then it went to the gamma state, the 8th state, and eventually it ends up in this alpha region. So what this is plotting, this is called a dynamic Ramachandran plot, and it is representing the conformational changes happening during protein folding. So the dynamic Ramachandran plot will tell us what are the different transition states that are there during the folding of the protein. That is, what are the different conformations the protein is going through before it is reaching the native state. So that way also the Ramachandran plot can give us an idea about the process of protein folding. And often Ramachandran map is used for model validation. So we'll talk about that. So in this structure, there are 286 residues, which are, so basically what model validation means is that suppose this is a protein, and using X-ray crystallography or NMR or even simulations, we have predicted this is the structure of the protein. But how do we know if this structure is the correct structure or not? For that, what has to be done is that for each amino acid, the phi has to be calculated and plotted on the Ramachandran map. We can see that 94.08% of the residues are falling within the allowed regions of the Ramachandran map. So this is a very good predicted model because most of the residues are within allowed region. Now, if we see that many residues are in the disallowed region, then we will say the predicted model is wrong. And then the experiment has to be repeated till we get a model where most of the regions are falling in the allowed region. So according to this result, this protein structure is highly valid because most of the residues are within the allowed region of the Ramachandran map. So like this, the Ramachandran map can be used to say whether a predicted protein model is valid or not. So there is something called super secondary structure also. Super secondary structures are often also called tertiary structures, but these we are specifically studying these because these are some of the ones that are found in nature. One is the helix-turn helix. It consists of two helices, one red here, one blue here, connected by a turn. And this is a motif that is often found in DNA binding proteins. Basically, one of the proteins recognize and bind to specific DNA sequence by forming hydrogen bond with the nitrogenous bases in the DNA. That is the recognition helix. And another helix is stabilizing the overall conformation by forming hydrophobic interactions with the recognition helix. So this motif, the helix-turn helix, is very often found in DNA binding proteins. Another kind of super secondary or tertiary structure is the 4-helix bundle. So you have 4 helices occurring at the same time, and in the inner part of the helices shown in green here, the hydrophobic amino acids are concentrated, so they are held together by hydrophobic interactions. And in the outer part of the helices shown by red, that is exposed to the solvent, all the hydrophilic residues are present so that they can be stabilized by interacting with the solvent. So these are called the 4-helix bundles. So these are the super secondary or tertiary structures which involve helices. There are structures which involve the beta sheets also. So you can have something called a beta meander, which consists of 2 or more consecutive antiparallel beta sheet. So you can see these two form one antiparallel beta sheet. These two form another antiparallel beta sheet. And so 2 or more antiparallel beta sheets linked together by beta turns is called a beta meander. And then you can have, so these are just one after the other. Now, instead of this, if you have a more complicated structure where these are also antiparallel beta sheets, but they are kind of folded. So here it is a bit complicated because the polypeptide is starting from this helix and then it is folding over itself into the last helix. So this kind of structure is called a Greek key motif. It consists of 3 antiparallel beta strands connected by hairpins, where the fourth is, so this is the, if we consider, so there is one short loop here, another short loop and one long loop. So this structure is kind of, these are both consisting of antiparallel beta sheets, but in one, the chain runs from one end to the other. And in another, the chain kind of begins from the middle and folds over and ends here. And you can have more complicated structures. We don't need to go into that, but they all consist. So here, basically, you have, again, you have this yellow is a beta strand and this red is a helix. So you can have alternating beta strand helix. So you can have structures like that, which is called a Rossmann fold. So these are different kinds of the supersecondary or tertiary structures. Again, some examples are shown here. We don't need to go into the details. Here, there are alternating beta strand and helix, which forms a kind of alpha beta barrel, where in the middle, the beta turns are, the beta strands are in the middle and the alpha helices are outside. And similarly, you can have other structures. We won't go into those right now. So these are all about the different kinds of organizations in proteins. Now we'll talk about the different interactions that are holding together these structures. So the strongest is the covalent bond, which with energies of around 90 kilocalorie per mole. But mostly the tertiary and quaternary structures are on non-covalent interactions, of which there are 4 types. The ionic bond of around 3 kilocalorie per mole, hydrogen bond and hydrophobic bond, both around 1 kilocalorie per mole, and the weakest interaction, that is the van der Waals interaction, of around 0.1 kilocalorie per mole. There are also the disulfide bond that we talked about, where 2 cysteine residues come close to each other, and there is oxidation, that is H is lost from both and the sulfur is connected, forming a disulfide bond. So this is another kind of the non-covalent interaction. Sorry, this is another kind of, this is a technically a covalent interaction. So it will fall within the covalent interactions. Now, in the ionic interactions, we know whenever there are positive and negative charges, they will form ionic interactions. Like if you have a basic residue like lysine, which has a side chain NH3 plus and a glutamic acid with a side chain O minus. They can form electrostatic interactions between the charges. And if there is an interaction between the hydrogen and the oxygen, that will be called a hydrogen bond. So like this, these are basically what lead to the tertiary and quaternary structures. Again, you can see here there is an aspartate with a COO minus charge and a arginine with a NH2 plus charged side chains. Different kinds of, so this plus can interact with this minus, this plus with this minus, this plus with this minus, and so on. So different kinds of such interactions, which are the electrostatic interactions, can form, which hold together an acidic and basic side chain. And the strength of these interaction depends on the charge and the distance between the amino acids, following Coulomb's law. And then you have the very weak van der Waals interaction. So we know that in an atom, in normal cases, the electron cloud is symmetrically distributed, so there is no net charge. But sometimes instantaneous transient dipoles can form. Like if the electron cloud is kind of displaced towards this side will get a net negative charge. And so this is a transient dipole. And what it will do is that because this side has a net negative charge, it will pull the nucleus of the adjacent atom close to it. And so this atom will also get a transient dipole. And then this minus charge and this plus charge will attract each other to form the van der Waals forces. Now, van der Waals forces are very weak. They are proportional to 1 by r to the power 6. So as you can see, so here there is a plot where in x-axis distance between atoms is plotted, and in y-axis the energy is plotted. So as the two atoms are coming close together, there is a certain point beyond after which the energy is stabilizing. So there is a stabilizing van der Waals force acting between the atoms. But after a certain point, where we saw during when we were seeing the Ramachandran plot also, that the repulsive energy becomes greater. So beyond this point, this is the ideal point where the energy is minimum, and this is the native bond distance that is formed. But if you force the atoms to come even closer, slowly the repulsive energy increases till the net energy becomes positive. That means it is not favorable for the atoms to come so close to each other. So the van der Waals and dispersion forces are proportional to 1 by r to the power 6, whereas the electrostatic forces are proportional to 1 by r square. So as r is decreasing, its effect on the van der Waals forces are much higher. You can also have something called hydrophobic interactions. In hydrophobic interactions, what happens is, suppose you have two polypeptides and water between them. Hydrophobic interactions, basically, the water will be removed and the two polypeptides will attract each other by hydrophobic interactions. So in this, there is certain enthalpic gain, because in the, so when this is happening, you have an enthalpic gain from hydrophobic interactions, and also because the water molecules that were trapped are being released, so there is an entropic gain also. So mainly the hydrophobic interactions occur in the interior regions of the protein. So you can see here, this is a protein. The green patches is representing a hydrophobic cavity. And in this hydrophobic cavity, a fatty acid, so fatty acid basically means a long chain alkyl group. So the long chain alkyl group can very nicely bind to the cavity of the protein, because in this cavity of the protein, there are hydrophobic residues. So like this, proteins can be designed which have different, so we saw in alpha helix, we can have one phase of the alpha helix that is hydrophobic, one phase that is hydrophilic, and so on. So we can design proteins also in such a way that there are certain patches of the protein that are hydrophobic, which are shown in green here, and they will be useful in binding to hydrophobic drug molecules like fatty acids, which are long chain alkyl groups. So that is hydrophobic interactions, where basically you have different alkyl groups, and they tend to aggregate with each other because when the water is being solvated, they cannot interact. But when this solvating water is removed, then they can interact with each other. And this is favorable because the hydrophobic interactions are providing an enthalpic stability, and the water molecule that is released, that is increasing the entropy of the system. So both enthalpically and entropically, this is favored. So hydrogen bond, we have already seen before, is that when we have hydrogen attached to an electronegative element, the hydrogen will have a delta plus charge, and it can bind electrostatically to a delta minus charge on an oxygen, forming hydrogen bonds. And hydrogen bonds are what is giving stability to the beta sheets. So whenever you have NH and CO close together, hydrogen bonds are forming. Both antiparallel and parallel beta sheet and alpha helix, hydrogen bonds are the main things that are giving stability to these helices. Now we will go into protein folding. So protein folding is the process by which a protein chain acquires its three-dimensional structure, which is generally the biologically functional structure. Now, in the folding of a protein, the delta G must be negative. So it must be thermodynamically favored. So in folding, what happens is that normally, if we look at it energetically, you see this arrow. This is representing the enthalpy of folding. So generally, when there is a free polypeptide chain and it is forming a secondary structure, you know, in secondary structure, there are hydrogen bonding interactions, dipole-dipole interactions, and so on. So in secondary structure, many stabilizing interactions are forming. So the enthalpy is favorable. That is, enthalpy is negative. But we also know that when it goes from a free structure like this to a folded structure, the entropy is decreasing. So delta S is negative. Delta S is negative. So if you have minus T delta S, that is positive. So entropy is unfavorable, enthalpy is favorable. So if you combine these two, delta G is delta H minus T delta S, you will have a net delta G of folding, which is around 5 to 15 kilocalorie per mole, and it is negative. So the folding process is thermodynamically favored. So basically, this is called the folding funnel, where you have many different states. So the protein initially starts, suppose it starts here. While folding, it will go into many different states. But so there can be multiple intermediates, but the native state is only a single structure. So it can fold along the folding process can occur in different ways, but all the process will eventually converge into one single native state structure. There was an experiment carried out by Christian and Anfinson called the ribonuclease refolding protein. Basically, ribonuclease is a protein where there are 8 cysteine amino acids forming 4 disulfide bonds. So each disulfide bond here is shown by a so between cysteine 26 and 84, there is one disulfide bond. Like that, between the 8 cysteine, there are 4 disulfide bond. Now, this protein ribonuclease is treated to urea in the presence of 2 mercaptoethanol. What this does, it will break all the disulfide bonds. So all the SS bond become SH-SH. So the disulfide bonds are broken and the protein is unfolded. After that, the urea and mercaptoethanol, which was causing the unfolding of the protein, that is removed, and the protein is put in a buffer solution. You can see that again the disulfide bonds have reformed. But the interesting part was that it is still forming the same structure. So you have a native structure, you have completely unfolded the structure, and then when you are allowing the protein to fold again, it is refolding to the same structure. It is not forming a different structure. So what this showed is that the native structure is common, which is what this funnel was showing, that no matter how the protein is folding, there is only one native state. That is what was proved through this experiment, that even after completely unfolding the protein, when it was allowed to fold again, the same structure was formed. So that was the thermodynamic hypothesis that said the native folded structure of a protein under optimal condition is the most energetically stable conformation. So always the most energetically stable conformation, that is the native conformation, is going to be formed. And also there is something called a kinetic theory of protein folding, which says that folding proceeds through different steps. but the protein does not try out all possible rotations of the conformational angles. So this is basically another way of what Ramachandran had found. That is what it is saying, that protein cannot try out all possible conformational angles because there will be some conformational angles that are sterically not available to the protein. And uh, so while the protein is folding, you can see that there are different intermediates, but there is only one native state. And sometimes there can be certain states which are very close in energy to the native state, but that is not the native state. So that leads to misfolding of the protein. So there are certain proteins like amyloid proteins which leads to Alzheimer's disease. Basically, while folding, if the protein enters this state, now in order to escape from this state and form the native state, a lot of energy is required. Sometimes the protein does not have that energy, so it gets trapped within this state, and that is when we say the protein is misfolded, and that is what leads to diseases. So instead of adopting the native natural state of the protein, if it gets trapped in a different state, that is called it is said to be kinetically trapped, and that is often what leads to diseases. And another problem that happens is that the protein is forming the native state, but some aggregation is happening. So aggregation means multiple protein molecules in the native state are coming together and they are forming an aggregate. So that is also a very unfavorable thing, and this also leads to several diseases. In order to prevent that, there are several biological mechanisms. We don't need to go into that, but this kinetic intermediate and the aggregate, these are two unfavorable things in the protein folding process that lead to different diseases. Now, protein folding itself, there are several models that have been proposed. One is the nucleation model, which says that some neighboring residues in the sequence form secondary structure. So you have a sequence, there is formation of secondary structure, like somewhere in the sequence helix is forming, shown in blue here. Somewhere in the sequence beta sheet is forming, shown in red here. And this secondary structure acts as a nucleus from which the native structure propagates in a stepwise manner. So you have a secondary structure forming here. So this region is like a nucleus. And around this region, the tertiary structure is forming, which leads to the native state. So the tertiary structure forms as a consequence of the secondary structure, and this is called the nucleation model. You can have another thing called the hydrophobic collapse model, which is saying that the protein will rapidly collapse around the hydrophobic side chain. So you have a protein here, it is just collapsing into so the nucleation model is saying that first secondary structure is forming, and then around the secondary structure, the different tertiary structures are formed. The hydrophobic collapse model is saying that first all the hydrophobic side chains like the leucine side chain, the isoleucine side chain, valine side chains, they are all interacting with each other to form a globular complex held together by hydrophobic interactions. And then the different hydrophobic interactions will rearrange to form the native protein. So these are two models that are proposed for the folding of the protein. There are several experiments that support the nucleation model and several experiments that support the hydrophobic collapse models. So basically, these are the two known mechanisms so far. And there are some enzymes that can be used to speed up the folding process. Some of these are peptidyl prolyl isomerases. So what they will do, we know that cis trans isomerization is a very slow process because the amide bond, peptide bond is a partial double bond. So the peptidyl prolyl isomerase will catalyze the cis trans isomerization, and hence it will help to speed up the protein folding. Then you have molecules like which are called molecular chaperones. They also help in folding of large proteins. So if you have a large protein that is interacting with other proteins, that interaction will prevent the protein folding. So what the molecular chaperone does is that it isolates the large protein so that it does not interact with any other protein, and hence it enables a large protein to quickly fold. So these are another is the protein disulfide isomerase, which helps in formation of correct disulfide bridges, which leads to formation. So we saw the ribonuclease experiment. So from this step to this step, if a protein disulfide isomerase had been added, that will help in quicker formation of the disulfide bonds, and so this step will be catalyzed. So these are different enzymes that speed up the protein folding process. And you can see that there are different kinds of proteins. Mesophilic protein is normal proteins which are found in the human body, where if you heat the protein, it will be completely denatured. So you can see this is the native structure of the protein. At high temperature, all the native structure is broken down and the protein becomes non-functional, that is, it becomes denatured. But there are certain proteins called thermophilic proteins. You know, there are some bacteria which exist in very high temperatures. So they have thermophilic proteins, which are stable even under high temperature conditions. So these thermophilic proteins basically have additional non-covalent interactions, which can be hydrophobic, they can be disulfide bridges or electrostatic. And these additional interactions increase the thermal stability of the protein. So even up to temperatures of 100 degrees Celsius, the protein does not denature when it is heated. It is still stable. So the bacteria which are found in natural hot springs or volcanoes, they have this kind of thermophilic protein where the number of non-covalent interactions holding the tertiary structure together are much stronger, and the number of interactions are also greater. Finally, there are some tests which we can use to identify whether suppose we are testing that whether a solution has carbohydrate or protein or what. So there are some tests which we can use to identify that protein is present in a solution. One of them is the biuret test. So suppose you have a peptide. So the peptide is represented by this one peptide bond occurring n times. So basically this is a long peptide. And if the reagent is biuret reagent is added, which contains sodium hydroxide, hydrated copper 2 sulfate, and potassium sodium tartarate. So the biuret reagent is added. Basically, the copper 2 plus ion forms a complex with the nitrogen lone pairs of the peptide bond. And this will lead to a deep purple color. But if the peptide bond is not present, like if you have carbohydrates or DNA, this complex will not form. And so whatever was the initial color, that will remain. So because of the formation of this complex, it can be estimated that protein is present. Another is the Follin-Ciocaltio assay, where again Cu2 plus ions are present and they are forming a complex with the peptide bond. And so that is giving an intense blue color. And if there are tyrosine, tryptophan, or cysteine residues present in the protein, that will further react with the reagent to give even another different blue color. So basically, again, we don't need to go into the chemistry of all these, how this is happening. The questions from here will be pretty simple. They can just say that biuret reagent is added to a solution of to a X solution, and no color and a deep purple color is seen. So what does that indicate? That the solution has protein. So these are basically qualitative tests for proteins. And there is another test called the Bradford assay, where again there is, so you have a dye called Comesi G250, and if it is added to a protein which has basic and aromatic side chains, there will be a color change from brown to blue. You can see here there is a series of test tubes where in the initial test tube there is no protein. So this is the initial brown color of the dye, and slowly the concentration of protein has been increased. So you can see slowly from brown there is a change to blue color as the concentration of protein increases. So if the protein concentration is plotted against the absorbance, a kind of increase can be seen from here, where initially the color was completely brown till you have 200 microgram per ml of protein, and the absorbance is high and the color is around blue. But the important thing to note is that for the so Bradford assay is a quantitative assay, because you can this is a from the curve you can estimate. So suppose we have an unknown solution and it is giving an absorbance of say 0.9. So we can draw a line and drop it to the x-axis, and that will give an estimate of what is the concentration of the unknown sample. So the Bradford assay can be used for quantitative determination of protein concentrations. Then there is something called ultracentrifugation, and it is a powerful technique for fractionating different macromolecules like DNA, RNA, and proteins. So how this is prepared is that you have a vessel where with a partition, and on one side you have a dense solution of sucrose, and on the other side you have a less dense sucrose solution, and they are properly mixed. The partition is raised and they are properly mixed. Then this is collected in a test tube. So in the test tube, the dense sucrose solution is settling at the bottom and the less dense is above it. To this, the macromolecule solution is added. It can be a solution of protein, it can be a solution of DNA. Whatever the macromolecule, a solution of the macromolecule is added on top. Then this tube is placed in a swinging bucket rotor. So when the rotor revolves, the tube moves to a horizontal position. So the tube was originally like this, and there is a rotor which is rotating the tube. So the what happens is that the tube is now horizontal and it is rotating in this direction. Rotor is rotating the tube in this direction. Then there will be a kind of separating out of this. So the most dense will be at the bottom. Then slowly as you go towards the top, the density will decrease. So now the sample has been separated out based on density, and you can collect the different. Suppose there is a mixture of proteins. The more dense protein will go towards the bottom and the less dense will be at the top. So using ultracentrifugation, you can separate out. One method of separating proteins we saw was the SDS page. Another method is the ultracentrifugation, which is separating the proteins by the size of the size and mass of the and density of the protein molecule. And there are certain methods which can be used to locate disulfide bonds. So one thing is first of all is how do we if we want to calculate how many methionine residues are there in a protein. Basically the protein is reacted with cyanogen bromide, and wherever there is a methionine is CH2CH2SCH3. Wherever there is a methionine, cyanogen bromide will react and it will lead to this kind of a cyclic structure called a homoserine lactone. So if there are 5 methionines, 5 such cleavages will occur in the, so you can see that cyanogen bromine is cleaving the protein here, and this part will form a methyl, will form broken peptide, and so you will get this will be one part of the peptide, this will be another part of the peptide. So cyanogen bromide is basically cleaving the peptide wherever there is a methionine present. So if you react a protein with cyanogen bromide and it cleaves into 2 parts, that means there is only 1 methionine present. If it cleaves into 4 parts, that means there are 2 methionine present, and so on. So it can be used to determine the number of, it can be used to cleave the protein at the methionine residues. These are basically different reactions that can be performed on the protein. And we don't need to understand why these reactions are performed, because that is outside the scope of this course. But we can just see that what are the different ways the protein can be manipulated. Another is, suppose you have a disulfide bond. The disulfide bond can be oxidized by performic acid to give, so it will break the protein wherever there is disulfide bond to form 2 cystic acid residues. Or you can reduce the disulfide bond by dithiothretol, and it will form 2 different, again the protein will be cleaved wherever there is a disulfide bond to form 2 SH residues, and then further reactions can be done. So these are different methods of cleaving the protein. One is cleaving them after methionine, and one is cleaving them wherever disulfide bond is present. And finally, why we are talking about cleaving proteins at all? This we studied in the first class. Edmond degradation is a method of determining the amino acid sequence in a protein. So in Edmond, we saw this in the first class, is that in Edmond degradation, the polypeptide is reacted with a phenyl isothiocyanate, and basically it will cleave the N-terminal residue. So if you have a polypeptide DEFGH, initially it will just cleave H. So you will get DEFG. Then it will cleave G. So you will get DEF. So like that, if Edmond degradation is done only once, you will get DEFG. If it is done twice, you will get DEF. And so on, you will get different fragments of the polypeptide. And by doing mass spectroscopic analysis of the different fragments, the protein sequence can be predicted. So that is why knowing how to cleave the protein is important, because only by cleaving the protein and then doing the mass spectroscopy of the different fragments, the protein sequence can be determined. And we saw in the last class that the amino acid sequence will help us predict that what is the different secondary and tertiary structures that will be adopted. And so finding out the sequence of the protein is quite important. And I think that covers all the lectures of week 2. So we'll quickly go into some of the practice problems. Basically, I'll be solving the week 2 assignment only. But while solving, we'll discuss also. So one question was that which amino acid is not chiral? We know the answer is glycine, because for chirality, the carbon needs to be attached to 4 different groups. And in glycine, we have the COH and amino group, but both the groups are hydrogen. So glycine is not chiral. All of the 20 amino acids, only glycine is not chiral. All the other 19 amino acids are chiral. So the correct answer is C. Then you have a question asking which natural amino acid has the highest molecular weight. This also I mentioned. This I mentioned. So tryptophan has one of the biggest side chains. So the answer of this is tryptophan. Then it is asking that what among these, which is the primary structure of the protein?
Protein sequence, I think.
Yeah. So alpha helix and beta sheet are both secondary structures, and super secondary is your tertiary structure. So the primary structure is just the protein sequence.
Yeah.
Then this is asking that if you have an alpha helix with 10 amino acid, what will be the height of the alpha helix?
13.5 or 15. I have doubts.
So see, you have to remember that For an alpha helix, per amino acid, the rise is 0.15 nanometer. So rise per amino acid is 0.15 nanometer. And the question is saying you have 10 amino acids. So 0.15 into 10 will be 1.5 nanometer, which is 15 angstrom.
15
So the
angstrom.
answer will be 15 angstrom. So you just have to remember this value. 0.15 nanometer is the rise per amino acid. Then the question is asking the most common residue found in beta turn is what?
Proline.
Yeah, and why that is happening is that you can see that in beta turns, certain Fisheye angles are required to be, so in the Ramachandran plot, wherever you have beta turn, these are the Fisheye angles that need to be adopted in order for the structure to be a beta turn. And because the proline is a cyclic amino acid, the Fisheye is approximately restricted to these values. Normally, if you have an acyclic amino acid, it can freely rotate, so the Fisheye can vary. But because proline has restrictions in Fisheye due to its cyclic nature, so it prefers to form beta turn. That is why proline is the most common residue found in beta turns.
Ma'am, where
Uh-huh.
where we see the turns in the beta turns, like beta sheets that turning, is there higher amounts of proline and glycine there?
Proline is more actually. I don't think glycine. So see, this is a beta turn, right? This is your side chain. So proline side chain is basically cyclic. So it is easier. So you can see why proline can be accommodated, right? It's a cyclic side chain. So wherever there is beta turn, occurrence of proline is very high. But glycine, I don't think like it has any preference like that.
Okay, ma'am. Okay.
Do you have some other question?
No, ma'am. No, ma'am. Please continue.
Okay, so this is asking that what is the dihedral angle?
Angle between two intersecting planes.
Yeah. So you can see we talked that dihedral angle is basically you have 4 atoms which constitute the dihedral angle. So the first 3 atoms is forming one plane. The next 3 atoms are forming another plane. And between these planes, you have an angle that is your dihedral angle. That is why it is the angle between 2 intersecting planes. Then this is the question that which residue can adopt Fisheye angles in all 4 quadrants of the Ramachandran plot?
It's glycine, I think.
Yeah.
Because in the previously you showed that.
Yes.
Yes.
So yeah. So glycine, I discussed that 18 of the 20 amino acids have one particular Ramachandran plot. The exception is glycine and proline. So because glycine has the least steric hindrance, because its R group is hydrogen, so it has the most area in the Ramachandran plot, which is allowed, and hence it occupies all the 4 quadrants. Then this is asking that which residue is involved in disulfide bond formation?
Cysteine, I think.
Yeah. So cysteine is where your side chain is CH2SH. So 2 cysteines, when they come close to each other, they will lose the hydrogen, that is, they will get oxidized, and the sulfur will be connected by a bond, which is called the disulfide bond. So cysteine is the only amino acid which can do this, because methionine also has a sulfur, but there is no hydrogen. The sulfur is attached to a methyl group. So the only amino acid which can form a disulfide bond is cysteine. Then this is asking that what is the DNA binding motif?
HLH. I think. Helix loop helix, no?
Yeah, so that also I discussed while I was discussing the different secondary structures. So helix loop helix is a very common motif that is found in DNA binding regions. And it consists of the recognition helix, which is the portion that binds to DNA. and one other complementary helix, which is helping to position so the complementary helix will just bind to the DNA backbone and position the polypeptide, and the recognition helix will recognize certain bases of the DNA and bind to it. So the helix, loop helix or helix turn helix, this is one of the most common motifs found in DNA. Another common motif is also leucine zipper, but that is not there in the options. So here your answer is helix loop helix.
Ma'am, what is, ma'am, please, what is SXXN motif?
Sorry, your voice is breaking.
Ma'am, what is the option C? SXXN motif.
Okay. Okay, I will write this down. I will explain. I don't have slides for this actually, so I will write this down and I'll explain in the beginning of the next class. SXXN motif, right? Okay, fine.
Okay.
Okay, then what are the models for protein folding?
It will be option D, all of the above.
Yeah. So different models. So you one is your nucleation, where secondary structures are forming, and from the secondary structure, the secondary structure is acting as a nucleus around which the rest of the tertiary structure is forming. Another is the framework model, which is initially similar like the nucleation model, that different secondary structures are forming. But the nucleation model is saying that from the secondary structure, the rest is growing. But framework model is saying is around the secondary structure, the rest of the model suddenly collapses very rapidly to form the native structure. So these two methods, the framework and the nucleation, the first step is similar, but in the nucleation, it is predicting that there is a steady growth, and in the framework model, it is predicting that there is a diffusion collision, meaning that these secondary structures, they will collide with each other and rapidly form the native structure. And hydrophobic collapse is kind of saying the opposite of framework. It is saying that first a collapse will happen because of hydrophobic interactions, forming a molten globule state. And from this collapsed globular structure, the secondary structures will form later in order to form the native state. So these are three different models of your protein folding, which are options A, B, C. So the correct answer is all of the above. This is another question, that naturally occurring proteins consist of what kind of amino acids? D amino acids or L amino acids, or both or none?
Ma'am, L amino acids.
Yeah. So this is also just a fact to remember. There is no reason or anything for that. So L amino acid is the one which is naturally occurring in humans. Then this question is asking that peptide group in protein has rigid planar structure. Which interactions is giving the peptide bond its partial double bond character?
We option D, resonance.
Yes. So that is correct. So like I discussed, this is the peptide bond. Now nitrogen can donate the lone pair and the carbonyl bond can break. So this will be the resonance structure. So these are two resonance structures. And so the peptide bond will exist in between these two structures. So there is a partial double bond in the structure. It is shown by one sigma bond and the pi bond is shown by dots, indicating it is a partial double bond. And because of this partial double bond, the peptide group is existing in a rigid planar structure. And this is resonance. So the option is D, which is resonance. Then it is asking, regular secondary structures such as alpha helices or strands of beta sheet are connected by a stretch of polypeptide that changes direction abruptly. What are these structures?
Ma'am, is it trans or beta bends?
Yeah,
Like beta
that is
turns?
correct. That is correct. So you can have, so like I discussed, whenever you have anti-parallel beta sheet, suddenly the strand cannot change direction. There has to be something causing this change of direction, and that is your turn or beta bend. Even between two helices, they connect two helices like this. So not only are they responsible for changing direction, but they are often connections between different secondary structures. So, and you can see the structure of the beta turn is such, your strand is coming in one direction, but after the turn, it is going in another direction. So turns or beta bends are what causes this change in direction. So the answer is D. Okay, then there is a question, which among the following residues is most likely to be present on the surface of a protein?
I'm not sure, ma'am.
Okay, I'll give you a
seems to me
hint.
like leucine.
No, I'll give you a hint. When there is a protein, the hydrophobic residues are preferred to be on the interior of the protein, and the hydrophilic residues are preferred to be on the surface of the protein. So in these 4 amino acids, which one is hydrophilic?
No, ma'am, I cannot remember that.
Okay. So basically, these are the structures. So you can see valine is hydrophobic. It has a alkyl side chain. Leucine also, it is hydrophobic. Isoleucine also hydrophobic. But arginine side chain has NH2NH. So it is a hydrophilic side chain. Now, when a protein is folding, it will always prefer the hydrophobic side chains, the red ones here, to be in the interior, and the hydrophilic side chains to be exposed on the surface. Because mostly the protein solutions are in polar solvents like water or methanol. So if the solvent is polar, it will prefer to keep the hydrophilic side chains on the surface so that they can favorably interact with the solvent. Now, in these 4 options, the only one which has hydrophilic side chain is arginine. So arginine is likely to be present on the surface of the protein.
Okay.
Yeah, so I think that's all I have. I will just, I gave my email address in the last class. Do you have that?
No, ma'am. Unfortunately, I don't.
I'll give it again. You can mail me with any doubts and I will clear the doubts at the beginning of the next class. So this is my email address. One doubt of yours I have written, which is the SXXN thing that I will clear.
Motif.
Other than that, if you
Yes,
have
ma'am.
any doubts, please mail me at. So this video and the slides will be uploaded. If you have any doubts, you just mail me at this address. And in the next class, before beginning the session, I will first clear the doubts.
Okay, ma'am. Thank you, ma'am. Thank you very much.
Okay, thank you.